Hi, I'm Nikolai Schwabe, CEO of ProImmune, and in this presentation I'll speak about the financial and business case of immunogenicity risk management. When we look at drugs that are currently in clinical development, it is noticeable that compared to even a few years ago, the landscape has changed. Now the majority of biological drugs in phase 2 through to market launch are drugs that are targeting existing rather than new mechanisms of action. As you can see from this chart, it varies by indication, but roughly 3 in 4 drugs target existing mechanisms. This means that the lion's share of the dollar value of biological drugs sold in the future is shifting away from first-in-class. Instead, best-in-class products will be the winners, not least from the perspective of the patient, including cost, ease of use, and side effects, including immunogenicity. Bearing in mind that the most common consequence of drug immunogenicity is treatment failure, it's important to realize that even a low percentage of treatment failure can cause the majority of the overall cost of treating a condition in the population. Payers will only become too aware of this. We should reflect on our experience of immunogenicity in the clinic. Immunogenicity is often only measured in clinical trials and in some cases in limited post-marketing studies as a formal requirement. We know that anti-drug antibody assays are often a challenge when patients actually have the drug in their serum and their antibody levels cannot be measured. Let's look at the $9 billion a year drug Humira, which was one of the leading treatments for rheumatoid arthritis. The label for Humira indicates moderate immunogenicity occurring in 1 to 12% of patients in clinical trials. However, there is now a publication out that indicates that more than 50% of patients in certain cohorts develop anti-Humira antibodies, and of those almost all develop reactivity against the binding domain of Humira. Although we do not have so much data for oncology antibodies yet, we would note that there is usually not much follow-up on ADA responses after the end of therapy with such drugs. So we have to ask ourselves, how much more effective could Humira and other biologics be if they had less immunogenicity, which in many cases may not yet have been properly documented? And likewise, if, in the future, there are better choices on the market for patients and payers, then they will find out about those and choose accordingly. Ask yourself this question: if you had rheumatoid arthritis and were about to go on Humira as a patient, knowing that there's a likelihood of more than 50% of you failing the treatment within 14 months due to unwanted immunogenicity, would you choose this drug or another one that is equivalent but only has a 5% neutralizing antibody rate, if such a drug were available? Would you regard that difference as important? Today there may be a limited number of options for patients, but we believe that most patients, their prescribing physicians, and their payers would consider the difference significant, and that this would play out as a very significant market share difference between Humira and our imaginary drug. We should also be mindful of other developments in the biological drugs market as this market starts to mature. We see a strong drive towards improved ease of use for patients, which includes formulating more drugs in pre-filled syringes, moving away from IV application. This in turn often requires high drug concentrations to deal with volume constraints. The route administration moves into more immunologically responsive compartments such as the skin or the mucosa. And the variability of the cold chain increases when drug application moves away from the hospital environment. We also see more and more innovation on containers, which in itself opens further complexities. The issue is that all these changes can affect immunogenicity adversely, and, being cautious, one has to assume that they will. I now want to switch gear completely and look at the financial parameters that drive the pharmaceutical industry. Looking at 3 large and representative companies that sell biopharmaceuticals, Pfizer, Amgen, and Biogen, we see a spectrum of turnover ranging from $5 to nearly $70 billion a year. On average, these companies spend 19% of their sales on research and development and have earnings or profits after tax of 21% of sales. I should say that these are plain averages and not weighted averages. Looking at the average value of these companies as a multiple of their earnings, we can see that a typical price to earnings multiple is around 20 times. I now want you to consider an example case of an imaginary company that has $10 billion turnover for normalization purposes. It would spend around $2 billion on R&D and earn $2 billion in profits after tax if it falls within the averages I just discussed. With the same PE multiple of 20 times, it would then be worth $40 billion. We assume that we have a typical R&D driven company that uses its own R&D to regenerate and grow its pipeline. Its R&D investment would require to replace its revenue every 15 years or so, as prior drugs go off patent. From our own experience, we assumed that supporting a $2 billion a year R&D pipeline with comprehensive immunogenicity risk management, using all appropriate assays when needed, to evaluate, rank and select drug candidates would result in an additional R&D spend of $15 million a year. Half of this would be internal spend and the other half with external vendors. And this would have to be carried on for 15 years, costing $225 million before it really starts paying back. So the question really is, what difference can an immunogenicity risk management program make to the financials? Well, in order to take a view on this, let's come back to the $24 billion a year anti-TNF market. Ablynx, a company in Belgium, has recently announced encouraging phase 2 results for a nanobody that could eventually directly compete with Humira. From the results, it seems that the potency of the drug is quite comparable to Humira, but it appears that this nanobody is inducing far less immunogenicity. The results are only from a phase 2 trial, but if the data in further clinical work confirms what is currently seen, and the drug progresses to market, it is credible that it could bite a huge chunk out of the anti-TNF market based on its advantageous immunogenicity profile alone. It will not be the same for every drug, of course. So now consider this. If our model company's research leads to drugs that compare favorably in immunogenicity over the competition, it may be reasonable to assume that this could increase the eventual revenue in its drug pipeline by, say, 20%. We have picked 20% as a pipeline average here, and sometimes the difference made to some programs may be negligible, while on others it may be decisive. If our assumptions hold true, then company revenue could eventually increase from $10 to $12 billion a year for this reason alone. Because this revenue shift is driven by competitiveness, it has little impact on running costs. The manufacturing and distribution expenses are usually relatively low. We can assume conservatively that around 50% of this revenue difference drops to the bottom line after tax. Let's look at this graph, which demonstrates how sustainable increases in earnings translate to added value. In fact, the $1 billion in added earnings results in $20 billion in added value. That's because of the price-earnings multiple, which is 20, and which we assume will remain the same. Hence, the 20% increased revenue produces a value shift of 50%. True, this will only take effect in 15 years, and over that time, some $200 million or so have to be spent on research in addition. But the nearly 100 times return on investment is enormous, and it is in fact so large that even if we are wrong in some of our assumptions, we have a very healthy margin of safety. We could be quite wrong and still be very right. Much of the industry is already performing immunogenicity risk management and have bought into the concept of de-immunized antibodies without perhaps realizing this. Chimerization, humanization, and fully human antibodies are effectively progressively de-immunized antibodies. One also has to bear in mind that developers have wholeheartedly adopted these technologies in the clinical development of drugs without knowing their effectiveness in advance. So why has everyone done this? It just made sense to do it conceptually. In fact, the success rate of these approaches is still not fully known, as not many of them have been tried in the clinic on a comparative basis. We don't know the outcomes of alternative scenarios. Few would say, however, that moving in this direction was unreasonable. In fact, as we will see later on, many of the results that have been obtained with these technologies correspond to general results obtained in T-cell assays. What we propose is that in addition to these conceptual design approaches, it makes sense equally to combine such approaches of including human sequence content with physical assays that can shed light on how effective such an approach turns out to be on a particular drug program before moving into the clinic. So let's look at evaluating T-cell responses and their relevance in this context. The underlying tenet is that anti-drug antibody responses do in the main require T-dependent help in order to become long-lived with high affinity. Assuming that this holds true, then a protein that has poor T-cell help is just less likely to be a strong immunogen. It is fair to say that this idea is in fact proved beyond doubt in the context of subunit vaccines and generally in immunology, where often wanted rather than unwanted immune responses are studied. Possible T-cell responses to a protein are actually easier to measure than possible B-cell responses at a preclinical stage. And we also propose that T-cell assays and other in vitro assays related to T-cell responses are good enough to get a good idea of the epitope content and antigenicity present in drug candidates. So what has been achieved or shown with T-cell assays so far? It is fair to say that mouse Fc regions are more immunogenic than human Fc regions in antibodies administered in humans, and T-cell assays reflect that. The same applies to human Fc allotypes when applied to recipients that do not have the same allotype. The same again could be shown for mouse versus human FV regions. As we will see later, humanized FV regions are demonstrably less immunogenic in the frameworks. The CDRs and back mutation regions of humanized antibodies in turn are often immunogenic, and also in humanized and fully human-derived antibodies, and again this is confirmed by T-cell assays. So in summary, the cumulative data available from T-cell assays today supports in each case what seems to be observed as immunogenicity from different sequence sources in the clinic. Let's illustrate this on a recent published example that we have worked on here at ProImmune. In this project, the sponsor, DSTL, who is part of the UK government, wanted to humanize an antibody for a potential passive immunization approach to protect individuals from Venezuelan equine encephalitis virus. An issue for the program was that the antibody, once made, could potentially not be tested in humans. DSTL had a good mouse antibody for their objective that they had already humanized using a standard approach. They now wanted to produce some assay-based evidence that indeed the humanized version would be significantly less antigenic than the mouse version. ProImmune compared the humanized and mouse FV regions of the antibodies involved, and we showed that 1. The CDR regions that remained unchanged did contain some significantly responding peptide epitopes, and 2. The T-cell antigenicity of the framework portion was significantly reduced as a consequence of humanization. Again, this result corresponded with what one would expect conceptually, and this could be viewed as an ex vivo validation study for the antibody design. So what can you do with this T-cell response-based information? As we have seen, it can be used to validate for individual projects the conceptual design approaches that are now commonly used in industry. We can select proteins from a group based on their observed antigenicity. The assay results can guide us in making changes to protein sequence if deemed necessary. To summarize then, we believe that immunogenicity is a product attribute that can be managed proactively. Engaging in such management will not be just an option for pharmaceutical companies in the future, but essential for ensuring that a drug will end up with an attractive competitive position in the market. Patients, physicians, and payers will choose the best product for their needs, and they will understand the importance of immunogenicity in this context. To manage a biological drug pipeline effectively in the context of this challenge, one cannot work piecemeal. Assays performed in isolation each time an individual question comes up on a program will only have a fraction of the effectiveness of a sensible assay program of complementary assays that is pre-planned throughout the development cycle. Working this way also saves a huge amount of cost in carrying out each individual assay and ensures that information is available to make the right decisions at the right time. And as we have shown with our financial workings, such a program can return a large multiple of the investment required, and with a significant margin of safety in the assumptions.